Welcome back to another exciting episode of Behavioral Neuroscience, lecture 22 on a really quite dreary Monday. Apologies for this once again being really too close to class time. This whole weekend and much of last week was taken up with grant preparation and submission and so on. Apologies if you don't get your questions in for this class exactly on time. I won't hold it against you. I hope everyone's holding up. I will definitely say that, as you can probably tell, my lack of haircut is really beginning to get to me. I'm going to be doing this through the whole talk because Anyway, that's not neuroscience. On to neuroscience. Picture on your screen. This is another one of the works from my favorite neuroscience artist, Greg Dunn. This is Gold Leaf. It's actually a picture of which I have a copy on the wall in my office. I paid for the original of this. And so this is the hippocampus, right? This is the curve from CA3, CA1 up to the dentate gyrus. You can see some fibers coming in here. These are mossy fibers coming in. These are pyramidal cells. The whole thing is done in gold leaf. I just think it's beautiful. Random news from the world of neuroscience. And this is really pretty up to date. This is a couple months ago. We've been talking about norepinephrine as a modulator of fear memory, and definitely in the basolateral amygdala, with the default assumption being that it's working on neurons, right? That's where the LTP is happening, that's where the end norepinephrine is going to be having its effect. This study really suggested that probably at least some of the effect of norepinephrine is actually on astrocytes. And so glial cells, never get enough attention. They do a whole bunch of stuff we don't really think about a lot of the time. I don't expect you to even try to understand this figure. The point is several measures of glial activity in response to multiple optogenetic manipulations. So like several recent papers, they've used a viral vector to encode a channel rhodopsin, specifically into, in this case, astrocytes. Excuse me. Excuse me, allergies, oh well. And they've confirmed that the vectors are expressed where they want to be by using fluorescent markers encoded in those same vectors. And then this whole slew of overly complex figures down here. This is basically just saying, hey, as it turns out, when you deliver norepinephrine, it's not just neurons that get activated, you also see increased activation of astrocytes. Calcium signaling goes up, some of the cyclic AMP, secondary messenger stuff. So there's definitely a role for astrocytes in this processing of fear memory in response to norepinephrine. So there's a modulatory role, not just for neurons, but for glial cells. We were talking last time a bunch about parasites. We're going to talk about that again next class. And in kind of preparation for that, I'm going to start off once again with somebody other than me talking. So this is a 13-minute video from Ed Yong. I talked about Ed Yong last time, one of the world's best science writers, really just an excellent voice for popularizing science without missing detail, without getting things wrong. He writes for The Atlantic. If you have a moment, go Google Ed Yong Atlantic, and there's a whole slew of really excellent columns, including, honestly, some of the best analysis of COVID-19 stuff I've seen. So 12 minutes of listening to him, not me.
A herd of wildebeest, a shoal of fish, a flock of birds. Many animals gather in large groups that are among the most wonderful spectacles in the natural world. But why do these groups form? Common answers include things like seeking safety in numbers, or hunting in packs, or gathering to mate or breed. And all of these explanations, while often true, make a huge assumption about animal behavior, that the animals are in control of their own actions, that they are in charge of their bodies, and that is often not the case. This is Artemia, a brine shrimp. You probably know it better as a sea monkey. It's small and it typically lives alone, but it can gather in these large red swarms that span for meters. And these form because of a parasite. These shrimp are infected with a tapeworm. A tapeworm is effectively a long living gut with genitals at one end and a hooked mouth at the other. As a freelance journalist, I sympathize.
You can tell that Ed is British. His humor may not quite convey. Oh well.
The tapeworm drains nutrients from Artemia's body, but it also does other things. It castrates them. It changes their color from transparent to bright red. It makes them live longer. And as biologist Nicholas Rode has found, it makes them swim in groups. Why? Because the tapeworm, like many other parasites, has a complicated life cycle involving many different hosts. The shrimp are just one step on its journey. Its ultimate destination is this, the greater flamingo. Only the flamingo can the tapeworm reproduce. So to get there, it manipulates its shrimp hosts into forming these conspicuous colored swarms that are easier for a flamingo to spot and to devour. And that is the secret of the Artemia swarm. They aren't sociable through their own volition, but because they're being controlled. Not safety in numbers, it's actually the exact opposite. The tapeworm hijacks their brains and their bodies, turning them into vehicles for getting itself into a flamingo. And here is another example of a parasitic manipulation. This is a suicidal cricket. This cricket swallowed the larva of a Gordian worm or horsehair worm. The worm grew to adult size within it, but it needs to get into water in order to mate. And it does that by releasing proteins that addle the cricket's brain, causing it to behave erratically. When the cricket nears a body of water, such as the swimming pool, it jumps in and drowns, and the worm wriggles out of its suicidal corpse. Crickets are really roomy, who knew? The tapeworm and the Gordian worm are not alone. They are part of an entire cavalcade of mind-controlling parasites, so fungi and viruses and worms and insects and more that all specialize in subverting and overriding the wills of their hosts. Now, I first learned about this way of life through David Attenborough's Trials of Life about 20 years ago, and then later through a wonderful book called Parasite Rex by my friend Carl Zimmer. And I've been writing about these creatures ever since. Few topics in biology enthrall me more. It's like the parasites have subverted my own brain. Because after all, they are always compelling, and they are delightfully macabre. When you write about parasites, your lexicon swells with phrases like devoured alive and burst out of its body. But there's more to it than that. I'm a writer, and fellow writers in the audience will know that we love stories. And parasites invite us to resist the allure of obvious stories. Their world is one of plot twists and unexpected explanations. Why, for example, does this caterpillar start violently thrashing about when another insect gets close to it and those white cocoons that it seems to be standing guard over? Is it maybe protecting its siblings? No. This caterpillar was attacked by a parasitic wasp which laid eggs inside it. The eggs hatched and the young wasp devoured the caterpillar alive before bursting out of its body. See what I mean? Now, the caterpillar didn't die. Some of the wasps seemed to stay behind and controlled it into defending their siblings, which are metamorphosing into adults within those cocoons. This caterpillar is a head-banging zombie bodyguard defending the offspring of the creature that killed it. We have a lot to get through. I only have 13 minutes. Now, some of you are probably just desperately clawing for some solace in the idea that these things are oddities of the natural world, but they are outliers. And that point of view is understandable because by their nature, parasites are quite small and they spend a lot of time inside the bodies of other things. They're easy to overlook, but that doesn't mean that they are unimportant. A few years back, a man called Kevin Lafferty took a group of scientists into 3 Californian estuaries, and they pretty much weighed and dissected and recorded everything they could find. And what they found were parasites in extreme abundance. Especially common were trematodes, tiny worms that specialize in castrating their hosts, like this unfortunate snail. Now, a single trematode is tiny, microscopic, but collectively they weighed as much as all the fish in the estuaries and 3 to 9 times more than all the birds. And remember the Gordian worm that I showed you, the cricket thing? One Japanese scientist called Takuya Soto found that in one stream, these things drive so many crickets and grasshoppers into the water that the drowned insects make up some 60% of the diet of local trout. Manipulation is not an oddity. It is a critical and common part of the world around us. And scientists have now found hundreds of examples of such manipulators. And more excitingly, they're starting to understand exactly how these creatures control their hosts. And this is one of my favorite examples. This is Ampulex compressor, the emerald cockroach wasp. And it is a truth universally acknowledged that an emerald cockroach wasp in possession of some fertilized eggs must be in want of a cockroach. When she finds one, she stabs it with a stinger that is also a sense organ. This discovery came out 3 weeks ago. Stabs it with a stinger that is a sense organ, equipped with small sensory bumps that allow her to feel the distinctive texture of a roach's brain. So like a person blindly rooting about in a bag, she finds the brain and she injects it with venom into 2 very specific clusters of neurons. Israeli scientists Frederick Libesat and Ram Gal found that the venom is a very specific chemical weapon. It doesn't kill the roach, nor does it sedate it. The roach could walk away or fly or run if it chose to, but it doesn't choose to, because the venom nixes its motivation to walk, and only that. The wasp basically unchecks the escape from danger box in the roach's operating system, allowing her to lead her helpless victim back to her lair by its antennae, like a person walking a dog. And once there, she lays an egg on it, egg hatches, devoured alive, bursts out of body, yada, yada, yada. You know the drill. Now, I would argue that once stung, the cockroach isn't a roach anymore. It's more of an extension of the wasp, just like the cricket was an extension of the Gordian worm. These hosts won't get to survive or reproduce. They have as much control over their own fates as my car. Once the parasites get in, the hosts don't get a say. Now, humans, of course, are no stranger to manipulation. We take drugs to shift the chemistries of our brains and to change our moods. And what are arguments or advertising or big ideas, if not an attempt to influence someone else's mind? But our attempts at doing this are crude and blundering compared to the fine-grained specificity of the parasites. Don Draper only wishes he was as elegant and precise as the emerald cockroach wasp. Now, I think this is part of what makes parasites so sinister and so compelling. We place such a premium on our free will and our independence that the prospect of losing those qualities forces unseen informs many of our deepest societal fears. Orwellian dystopias and shadowy cabals and mind-controlling supervillains, these are tropes that fill our darkest fiction. But in nature, they happen all the time. Which leads me to an obvious and disquieting question. Are there dark, sinister parasites that are influencing our behavior without us knowing about it, besides the NSA? If there are any, got a red dot on my forehead now, don't I? If there are any, this is a good candidate for them. This is Toxoplasma gondii, or Toxo for short, because the terrifying creature always deserves a cute nickname. Toxo infects mammals, a wide variety of mammals, but it can only sexually reproduce in a cat. And scientists like Joanne Webster have shown that if Toxo gets into a rat or a mouse, it turns the rodent into a cat-seeking missile. If the infected rat smells the delightful odor of cat piss, it runs towards the source of the smell rather than the more sensible direction of away.
If you have a moment, Google, Toxoplasma gondii mouse behavior on YouTube. There are some great videos of mice charging towards cats, jumping up on them, and the cats fleeing in kind of bemused terror. You wonder what the hell this mouse is doing. It's really kind of fun.
The cat eats the rat, Toxo gets too upset. It's a classic tale of eat, pray, love. We're very charitable, generous people. Hi, Elizabeth, that's short. Right, how does the parasite control its host in this way? We don't really know. We know that Toxo releases an enzyme that makes dopamine a substance involved in reward and motivation. We know it targets certain parts of a rodent's brain, including those involved in sexual arousal. But how those puzzle pieces fit together is not immediately clear. What is clear is that this thing is a single cell. This is no nervous system. It has no consciousness. It doesn't even have a body, but it's manipulating a mammal. We are mammals. We are more intelligent than a mere rat, to be sure. But our brains have the same basic structure, the same types of cells, the same chemicals running through them, and the same parasites. Estimates vary a lot, but some figures suggest that 1 in 3 people around the world have Toxo in their brains. And typically, this doesn't lead to any overt illness. The parasite holds up in a dormant state for a long period of time. But there's some evidence that those people who are carriers have score slightly differently on personality questionnaires than other people, that they have a slightly higher risk of car accidents, and there's some evidence that people with schizophrenia are more likely to be infected. Now, I think this evidence is still inconclusive, and even among toxic researchers, opinion is divided as to whether the parasite is truly influencing our behavior. But given the widespread nature of such manipulation, it will be completely implausible for humans to be the only species that weren't similarly affected. And I think that this capacity to constantly subvert our way of thinking about the world makes parasites amazing. They're constantly inviting us to look at the natural world sideways and to ask if the behaviors we're seeing, whether they're simple and obvious or baffling and puzzling, are not the results of individuals acting through their own accord, but because they are being bent to the control of something else. And while that idea may be disquieting, and while parasites' habits may be very grisly, I think that ability to surprise us makes them as wonderful and as charismatic as any panda or butterfly or dolphin. At the end of On the Origin of the Species, Charles Darwin writes about the grandeur of life and of endless forms, most beautiful and most wonderful. And I like to think he could easily have been talking about a tapeworm that makes shrimp sociable or a wasp that takes cockroaches for walks. But perhaps that's just a parasite talking. Thank you.
So welcome back to Parasites. Next class, in fact, we're going to talk more about consciousness and what it means to be in control of yourself. Things I've been mentioning as kind of side thoughts throughout the semester. We'll come back and consider this whole thing in more detail next class. Other bits to round up, loose ends. We were talking also last time about memories, and some of the questions in class and so on were about, well, can you really implant a genuine memory? Is this ecologically valid as opposed to being something you can just do in the lab? There's a really nice study about zebra finches. These birds need to learn a particular song. If you manipulate their memory centers with optogenetics, so standard methods, expressive channel rhodopsin, modulate the vocal modal pathway in the finch where that memory gets learned and stored. You can then mess with that pathway and you can persuade these zebra finches to learn a song that isn't the song they would usually learn. And in fact, as it says, they can override other individuals' song inputs, but it doesn't prevent them from subsequently learning other songs. You're not destroying anything. You're just telling them, hey, by the way, activate this pathway, learn this particular fake song, this fake memory we are introducing into your brain so that you have in a finch sense, a memory of having learned a really important song that never actually happened. You can directly stimulate these sensory motor synapses in the pathways that would be involved in songbird learning to create the memory, and you can show this because the bird will go off and sing this song, try and attract a mate, to create the memory of having learned a song that never existed. So yeah, you can absolutely, at least in a zebrafinch, implant a specific memory of something that never occurred at all. It wasn't in a different environment, just didn't happen. We spent most of the last time talking about bad things that can happen to the brain. Tapeworms eating holes in it, tumors causing pressure inside the skull, strokes causing apoptosis, this programmed cell suicide, because when you have too much calcium coming into a cell, and that's a consequence in turn of lack of control of membrane potential. My brain's going, too much coffee. When you have too little glucose, cells can't maintain membrane potential. That means they release glutamate. That means that more calcium can come in through NMDA receptors. That unregulated calcium entry triggers this pathway leading to activation of caspases, the enzymes that cause cell death, apoptosis. So that's the impact of a stroke or any interruption of fuel supply. You can imagine perhaps a tumor that grows and blocks a blood vessel, that would actually cause apoptotic cell death. If you get an infection, whether it's bacterial or viral, you can get meninges. These meninges can swell, and so they can cause meningitis, a swelling of the meninges, looking very much like basically a small brain tumor across the whole surface of the brain, squishing it down. You can get infection by viruses like rabies. You can get prions, these wonderfully scary infectious proteins that don't need any DNA, don't need any RNA. The protein itself causes other proteins to misfold, causing diseases like BSE. The bovine spongiform encephalitis is basically a variant of CJD, Creutzfeldt-Jakob disease. These two horrible conditions both cause you to get holes in the brain. And then we can treat some of these things. So we started talking about protein misfolding in the context of diseases like Parkinson's and Alzheimer's and so on, and ways you can treat that. You can give drugs like L-DOPA, you can give stem cells, you can have deep brain stimulation. Protein misfolding is a common characteristic. So this problem of abnormal protein degradation in Parkinson's, where you don't have that pathway that breaks down abnormal folding. Things like beta amyloid and tau building up in the Alzheimer's brain. You end up with these characteristic plaques, clumps of amyloid outside the cell, misformed and shrunken, shriveled neurons because tau, the protein inside a skeleton, is not working correctly. And those are all linked to abnormalities in cellular metabolism. So what I'm going to spend most of today talking about is actually some of my lab's work, looking at the links between things like brain metabolism, beta amyloid deposition, brain insulin signaling, those kind of variables. And you can see here, the areas in the brain where you get buildup of beta amyloid are the same areas where it turns out you're going to get impaired brain metabolism. Usual question pause. I guess this slide's almost here as like a marker for myself of previous times, but it also serves right as the illustration of that stuff we've talked about previously. So that should have been review. Now we're going on to new stuff. One side note, I guess, we talked about deep brain stimulation in the context of Parkinson's, and I mentioned it, is increasingly used for all kinds of things, losing weight, giving up smoking. The incidence of basically selective or optional deep brain stimulation surgery is really going up a lot. So it's very effective in many cases. It's being increasingly commonly used, but not just for really serious conditions like Parkinson's, but also for more essentially minor things. Giving up smoking is great, but having brain surgery to do it is a pretty drastic approach. Here's an example where deep brain stimulation in the Alzheimer's brain is being used. So individual MRIs, as you'd expect. The fornix is the input path to the hippocampus. And you can see here, post-surgery, here's the track of the electrode that's being put in to stimulate that. So basically these patients are getting increased input into the hippocampus. Here's the individual patients. You can observe, the tracks of these electrodes. These are pretty deep down into the brain. These are not small surgeries that are going on. Did it work? The answer is yeah. So the many mental status exam we've encountered previously, you probably recall, it's a very simple cognitive test. What's your name? What state do you live in? How many children do you have? That kind of thing. As you get changes in that score, an increase in change means more errors. And so that's a bad thing. So these are the 6 patients in the study. And you can see 5 of them over the preceding year had got worse on that scale. The last one basically stayed the same. After the surgery, after deep brain stimulation, each one of these patients got less bad, right? So a couple of them improved. This guy who had been going downhill 9 points on the scale in the previous year only went down by 5 points. These two both got better. This one last patient actually got worse a little bit. So 5 out of 6, the operation worked cognitively. And what was really interesting was they had pre-operation this characteristic loss of metabolism. So blue here is low brain metabolism compared to age-matched controls. And after that deep brain stimulation, 1 month later, even 1 year later, these areas of red are increases. So very long-term effects of maintaining an increase in brain metabolism, particularly in the areas that had been decreased, also in some other areas like the cerebellum, as a consequence of this hippocampal stimulation. So basically, DBS, deep brain stimulation, for Alzheimer's disease also seems to work. It hasn't been followed up very much. I'm kind of surprised that there aren't more patients with Alzheimer's getting these deep brain electrodes. It's not clear to me why that hasn't clinically taken off as much. We ended last time beginning to get into some of the links between insulin resistance and Alzheimer's, one of which, is caffeine. So caffeine is definitely neuroprotective. There was a study out just this week that suggests I may be giving myself not quite as much help as I would like, because it's possible that filtered coffee is better for you than the unfiltered French press stuff I drink. This may have too much cholesterol as a result of the coffee oils. Nonetheless, caffeine is definitely neuroprotective, probably against Alzheimer's and Parkinson's. And in this case, there was a rabbit model. And basically what they did was give animals either a control, relatively low-fat diet, or a high-fat diet, high cholesterol. And the brown stain here shows you gaps in the blood-brain barrier. Their results were really quite striking, as you can see. animals that didn't get caffeine on this high cholesterol diet had a fairly leaky blood-brain barrier. And that was entirely reversed by caffeine. So giving the caffeine treatment appeared to protect against some of the development of Alzheimer's disease. That suggests, and there are various lines of research coming together here, that perhaps things that help with type 2 diabetes, so insulin resistance, not type 1 diabetes, where your body doesn't make insulin, but type 2 diabetes, where you have too much insulin, generally linked to obesity, lack of exercise. Drugs that improve that insulin sensitivity, that metabolism, might also help for treating Alzheimer's. And so that's where we start, right, with the talk about my work. So the rest of today is going to be a discussion of things we do in the lab. And this really is the accurate subtitle: Save your brain by going to the gym. One of the best things you can do, several of the best things you can do to help your brain, help your cognitive function, you might not think of as having anything to do with the brain. So going and working out, really important. Getting good sleep, really, really important. We'll come back to that at the very end of the talk. Diet type stuff, all these things that affect this whole bag of flesh you're walking around in. Because the point is, right, we are not just brains in a vat. You know, many psychologists maybe might want to think of it that way, but we are really genuinely not just some kind of psychological phenomenon off by itself. We are biological basis of bags of flesh. And so, yeah, fine, maybe we think of ourselves as being kind of shadows of different, I don't know, psychological impulses hooked up somewhere in the matrix. You're not. You are a brain in a skull in a body. You are a biological organism that happens to be driven around by this brain, but the brain is not all of you. And so conditions that affect your body also affect your brain. That's the key point here. The condition we're going to talk about today is brain metabolism as regulated by insulin signaling, beta amyloid, Alzheimer's disease, diabetes, that kind of stuff. And so these two basic molecules, insulin is the canonical blood glucose regulator. As we've seen, it gets glucose out of the blood into cells. Beta amyloid is the canonical Alzheimer's protein. So we've seen it builds up and it causes those plaques in the Alzheimer's brain. And they both modulate glucose. They're both able to regulate brain metabolism. They both cause changes in brain metabolism when they are given either short-term or long-term. And so we're going to talk about a bunch of studies that implicate glucose metabolism as being a key player in these two effectors of disease states. We'll talk a little bit about caffeine at the end. But let's go back and look first and think about some history. Mid-1970s, there is a bunch of work, particularly driven by Louis Sokolov. Louis Sokolov is a totally brilliant guy, develops this method called 2DG, which we saw way back in methodology, allows you to look at brain metabolism in the awake animal, and the animal is doing something or seeing a visual pattern and so on. And we get a model built on some of those data of brain glucose supply. The problem with that, in part, was that we knew psychologically that giving extra glucose modulated memory and made it better. But the model of brain glucose supply suggested there was plenty of glucose already around. Well, how could this be? We knew that if we gave epinephrine, so adrenaline, this is actually some of the work from my doctoral advisor, Paul Gold, back when he was a graduate student, that you get this nice inverted U dose response curve. So we've talked about this. A little bit of stress, a little bit of epinephrine makes you smarter. Too much, and the signal to noise ratio goes back down again. Basically, it's almost like you're having a small seizure. You're overexcited, and so you don't get any better. Epinephrine doesn't get to the brain. What it does do is release peripheral glucose, and so the same researcher, Paul Gold, looked at glucose directly. These are animals being given a dose of glucose just after training on a memory task, and then the memory is being measured the next day. Again, nice inverted U. There's definitely a dose of glucose that works well. Too little, too much, no effect. Glucose does get to the brain. Glucose is obviously the fuel your brain demands. So we had this nice set of psychological data saying, hey, yeah, glucose makes you smarter. And a brain glucose model that said, there's always plenty of glucose. What are you talking about? That was where my graduate work basically came in. And so I developed a method, you've seen this in the paper, this is McNay et al. 2000, to look at these dips in brain glucose and showed, no, there really isn't an excess of glucose supply. In fact, in the basic rat brain, and this is true in humans too, if you put the rat in a complex maze task, this gray bar, glucose in the brain areas that are responsible for modulating that task gets drained. It's only if you give the rat a little extra glucose they get smarter, and you don't see that drainage. And so this links very nicely, of course, into conditions like Alzheimer's. The areas in the brain that show cognitive impairment. Alzheimer's patients have very poor memory. The brain memory areas also show impaired glucose use. If you give an Alzheimer's patient a little glucose, they do in fact get significantly smarter for the next half an hour or so. That was directly shown by, yeah, it's that guy again, Paul Gold. So he's going to show up a bunch in the early work here. Glucose and memory is really his field. This was a study I was actually around for, published in '93. So just before I arrived in graduate school, I did some of the follow-up work. Alzheimer's patients definitely respond to glucose. So do people with age-related memory deficits. So do people with Down syndrome. Several classes of people where brain glucose metabolism is impaired all show cognitive enhancement if you give them extra glucose. All right, so when I left my graduate work, I went to do a postdoc over at Yale, and I was really interested in factors that affected brain metabolism. One of the obvious ones seemed to be insulin. And this again was a case where there was a dogma in the field that just didn't seem very likely to me. The dogma was the brain doesn't respond to insulin, that the metabolic regulation inside the blood-brain barrier wasn't insulin sensitive. Insulin was only a blood peripheral hormone. That was really at odds with the fact that you could find insulin machinery within the brain. Now, there are other molecules, insulin-like growth factors, that can activate some of that insulin machinery. It just seemed more plausible that insulin was going to be working within the brain. And so we started to look at that stuff. Around the same time, a couple of other people, Suzanne Kraft in particular, were doing work looking to see, could the brain respond to insulin? This study, honestly, is a really terrible study, but it got the field thinking about it. She gave a whole bunch of extra insulin into Alzheimer's disease patients. And so, yeah, they improved their cognition. The study was massively confounded by the fact they also had like 10 times as much glucose in the brain. The point was the field started thinking about brain insulin responsiveness. One of the best studies, and I say this even though it's my study, we published in 2010. And what we did was directly block insulin in the hippocampus. So we gave a small antibody-like fragment. This is a little bacterially derived peptide. And what it does is bind to insulin and prevent it from acting. This is actually a therapeutic approach that's being taken increasingly now. A lot of the drug adverts you'll see on TV, the drug name ends in AB, which is an antibody. So these are biological drugs. They're basically antibody fragments that mop up a particular signaling molecule. We did exactly the same thing, except against insulin, directing it directly into the hippocampus. And what you see when you do that is the performance goes down chance levels. If you just block insulin signaling in the hippocampus, you don't give any extra insulin, you just block insulin that might be working there, you cause this marked memory impairment right down to chance level. If you give a similar peptide that acts against, this is actually ERB2, which is a breast cancer protein, it's a control peptide, does nothing as you'd expect. So blocking insulin within the hippocampus markedly impaired memory. And conversely, that same paper, we showed that if you give insulin in the hippocampus, you get this really nice inverted U. It just looks just like you gave them glucose or norepinephrine or any of those things. These white bars are all control animals. They're fed a healthy diet, they're not particularly fat, they're just kind of regular lab rats. If you take a bunch of animals, standard lab rats, and you give them, instead of a fairly healthy control diet, you give them a very high fat, high sugar, high calorie diet. Some of those rats act as though they're watching their waistline. They adjust their intake so they don't eat any more calories, they don't get obese, they look perfectly healthy and fit. We call those guys diet resistant because they resist the effects of this high calorie diet. And then when you test them with insulin, they look just like control animals. They're perfectly healthy and good. Some rats, conversely, you give them this high fat, high calorie diet, and they're basically like, yum, Twinkies, and they chow down, they get markedly obese, they develop insulin resistance. It's the model we use of diet-induced type 2 diabetes. It has many features in common with human type 2 diabetics who eat too much, become obese, develop insulin resistance. And those guys are markedly impaired. They have markedly impaired spatial memory here, both at baseline, so without any treatment, this is artificial extracellular fluid, it's a vehicle. Those guys on the diet resistant group are markedly better than their colleagues who've chowed down too much become obese. Obesity is linked to cognitive impairment. That's true in humans as well. Then if you look to see at these other doses of insulin, a dose of insulin that enhances memory in healthy rats doesn't do anything for the high-fat, insulin-resistant guys. You've got to give 10 times as much to kind of bash them over the head with this insulin hammer and make them get smarter. You can do it, you can enhance, but they're insulin-resistant in the brain. If you look to see what's happening metabolically, you drip a little insulin in the hippocampus, and then you look to see what's happening in the extracellular fluid, so the space outside the neurons. As you might expect, when you start delivering insulin, glucose goes into the cells, so the level outside the cells goes down. That's what insulin does to muscle cells, what it does to fat cells, does it to neurons as well, or at least to cells in the hippocampus. Could be neurons, could be glia, right? So glucose is being moved into the cells. And at the same time, you see this rise in lactate. Just like in a muscle, when you exercise and work out, you produce lactate. You burn glucose, you produce lactate. The fact that we're seeing this increase in lactate here means this glucose that's being pushed into the cells is being used. They're burning it up, they're producing energy, they're giving out lactate as a waste product. So you give a little insulin, the hippocampus revs up the metabolism. You basically boosted the engine. If you do the same thing though, in animals with that diet-induced resistance, nothing happens. And so what you see is that these animals don't have that same level of insulin stimulation. Instead, they show nothing at all. So as well as being cognitively resistant, as well as not showing the cognitive enhancement by insulin, they also don't show a metabolic enhancement. So the hippocampus is markedly insulin resistant. Incidentally, if you look at this initial rise, the initial rise in lactate is faster than the drop in glucose, and that actually has to do with glycogen. Insulin appears to increase the use of glycogen, the brain's energy reserve, very early on. So there are several things that are going on in response to insulin. But the basic concept that insulin is increasing local glucose metabolism is absolutely correct. And I wrote a review paper 2011 with one of my graduate students, and it basically just says, look, the brain definitely responds to insulin. It is still true now in 2020 that many of the med school textbooks still say the brain doesn't respond to insulin. I get texts from my undergrads who've gone off to med school saying, they're still telling us this. If anyone tries to tell you that, they're wrong. The brain is clearly regulated by insulin. So things that can make you smarter, right? I told you this, good diet, a little tiny, small amounts of stress, some good exercise. If you don't get those things, if you eat poorly and you don't sleep, you don't work out, and you're chronically stressed, so you're getting those glucocorticoids killing off your brain cells, all of those are going to cause cognitive impairment. It's really pretty bad. Number one, go to the lab. Go to the gym. Good grief, I need more coffee. I apologize. All right. If all of this is true, right? If we think that we are getting a link between insulin resistance and potential longer-term consequences like Alzheimer's disease, do we see an increase in amyloid beta, for instance, with insulin resistance? So there are several studies building on this initial work about insulin helping Alzheimer's disease. The first study that really kind of lit a spark under this particular idea, 1997, showed that one of the major risk factors for developing Alzheimer's disease is type 2 diabetes. If you have type 2 diabetes, your risk factor for developing Alzheimer's disease goes up anywhere from 2-fold to as high as 8-fold. It varies a little by study. It probably varies a little by population and by country and by diet and by lots of other variables. But all studies show the same thing. Your risk factor for getting Alzheimer's is much greatly increased if you have pre-existing type 2 diabetes. This is certainly true in terms of competition between the two molecules. Insulin and beta amyloid, it turns out, actually compete for certain breakdown pathways, and we'll come back to that. There are many pathways now identified that link insulin signaling to a bunch of the different outcomes that you see in Alzheimer's disease. So several stages of things that happen in response to healthy insulin signaling. If you block those, you cause Alzheimer's-like symptoms. Not only that, there are some complex links between insulin and beta amyloid processing. And so the basic story we're getting to is that insulin and beta amyloid interact with one another at several levels. If you look in transgenic models of Alzheimer's disease in mice. These are animals that chronically overproduce beta amyloid. And you put them on a high-fat diet, you see a great increase in that amyloid accumulation. So again, suggesting high fat, obesity, type 2 diabetes, likely to make Alzheimer's disease more likely and likely to make it worse. Here's the basic model that we ended up with, and I stole this from a colleague in University of Dundee. In the healthy brain, you've got some insulin, you've got some beta amyloid. You know, we talked last time, beta amyloid isn't just there to give you Alzheimer's disease, it has a healthy role to play in things like inflammation. Both those molecules, both insulin and beta amyloid, get broken down by this same enzyme. It's called IDE, insulin degrading enzyme. Gives you a pretty clear idea of what it does. It also breaks down beta amyloid. So in the healthy brain, there's plenty of IDE to go around. Insulin breaks down, A beta breaks down, insulin signaling is perfectly fine. All this stuff downstream of insulin signaling, things like GSK3 beta and PI3 kinase and so on, you don't care about details here. What you care about is that insulin signaling is definitely active within the brain, and it will lead to, for instance, increased glucose metabolism. During hyperinsulinemia, during the condition where you have type 2 diabetes or you're insulin resistant, one of the consequences of your brain and your body being insulin resistant is that they adapt. They make more insulin to try to overcome that insulin resistance. That extra insulin ends up taking up all of the capacity for breakdown of IDE. As a consequence, A beta amyloid here, doesn't get broken down. All the insulin degrading enzyme is breaking down insulin. And so amyloid beta builds up. It actually directly blocks insulin signaling. And so now all that healthy increased metabolism and cascades that happen downstream of insulin, those don't happen. So you have a direct molecular link between insulin resistance, obesity, hyperinsulinemia, and accumulation of beta amyloid, which then in turn further decreases insulin signaling, and you get this vicious spiral. There are several other points where insulin and beta amyloid interact. I told you insulin regulates amyloid precursor protein. So normally, this kind of squiggly thing here, this is the amyloid precursor protein, the protein that turns into beta amyloid. As it moves from the endoplasmic reticulum out to the Golgi apparatus, the trans-Golgi network, kicked through the plasma membrane and out of the cell, these A beta fragments, these beta amyloid fragments, that whole process is regulated by insulin. If you have impaired insulin signaling, beta amyloid, it builds up abnormally within the cell. Conversely, beta amyloid itself binds to insulin receptors. So this is a picture from a cultured neuron, and the little dots of light are fluorescent tags for insulin binding an insulin receptor. Over here, if you wash that cell with beta amyloid and then see whether insulin can bind, you can see it's markedly reduced. So C and D are little sections from the two cells. The ability of insulin to bind is greatly diminished by beta amyloid. You see the same thing again over here. Beta amyloid directly competes at the insulin receptor. And so when you get too much beta amyloid building up, you get this impaired insulin signaling. And conversely, if you give a bit of extra insulin as a small dose, it prevents the binding of beta amyloid. So the two molecules are competing at the same binding site, right? Net result, these two proteins oppose each other in the brain. They have opposite effects. Insulin increases metabolism, beta amyloid decreases metabolism. Great. So we have an explanation perhaps for why being type 2 diabetic increases your risk for Alzheimer's disease. What happens if we give, insulin? Well, we've shown you that it works in humans. There are now several studies using intranasal insulin to confirm that yes, insulin affects the brain, improves cognition, appears to modulate beta amyloid. And so there are several clinical trials that attempted to use that as a therapy for Alzheimer's disease. Reason it doesn't work. Think about the situation in the brain of these patients. They are likely type 2 diabetic, so insulin resistant. Particularly in the brain, there's really good evidence that even Alzheimer's patients who aren't type 2 diabetic in their body may be insulin resistant in the brain. And so actually some people now refer to Alzheimer's disease as in some cases type 3 diabetes. You get diabetes of the brain in particular. So they've got this abnormal insulin buildup. You give a little extra insulin, It pushes the beta amyloid away from the insulin receptor for a moment, and sure, has a short-term benefit. But if anything, it's going to make that insulin resistance worse. And so the long-term chronic effect is actually likely to be that you make the disease worse. If anything, you cause more amyloid accumulation. And that's what was found in clinical trials. None of the attempts to use insulin as a therapy in Alzheimer's have worked. If all of that's true, and there's no reason to doubt it, we wanted to know, well, wait, if insulin and beta amyloid are interacting so acutely, and we know that insulin is a cognitive enhancer, what about beta amyloid? Can we cause acute cognitive impairment? We think of beta amyloid as being this long-term build-up type drug. Well, does it work in the short term? If it blocks insulin receptors, does it cause impairment? And the answer is yeah, right? So when you give beta amyloid directly into the hippocampus, I don't know why this little significance asterisk has gone dark and dark blue, but you see this marked impairment. Beta amyloid 10 minutes later causes marked spatial memory impairment. And if you look at glucose in the hippocampus of those animals, the green line basically replicated our previous work. We showed that yes, putting the animals on a maze does again cause this dip, this drainage in extracellular glucose. Beta amyloid entirely blocked that, completely prevented the hippocampus from turning on, prevented it from using glucose, caused memory impairment. So if beta amyloid is doing that, what's the mechanism? Is it via glucose transport? There are two kind of canonical baseline glucose transporters in the brain, GLUT1, GLUT3. Don't worry about the details. The basic answer is no. Beta amyloid was doing nothing to those. But there's also this third kind of glucose transporter that gets ignored some of the time. That's called GLUT4. GLUT4 is the insulin-regulated glucose transporter that we've seen gets glucose out of the blood into muscle and fat cells. Many of the pathways that are downstream of insulin signaling in the brain also regulate GLUT4. And what GLUT4 does is it goes up to the cell surface. It's regulated by many molecules that are known to affect memory. And it turns out to be expressed specifically in the hippocampus, also in the cerebellum and the olfactory bulb. But the expression in the hippocampus is really interesting. In the same brain region where you see cognitive impairment caused by beta amyloid, where you see cognitive challenges in type 2 diabetes, you've got this glucose transporter that responds to insulin. And in most places in the brain, it's not there, but in the hippocampus it is. If you look in the brains of diet-induced obese animals, these animals that show high-fat diet caused insulin resistance, there's less of that glucose transporter at the cell surface. They have the same total amount, but it's not at the cell surface helping glucose to get into the cells. Okay, that's consistent. Interestingly, if you just take some healthy animals and you put them in a maze task, and then you look in their hippocampus, what you see is that the task itself causes those GLUT4 glucose transporters to go up to the cell surface. The task is demanding, is making them want glucose, and their brain is responding by moving GLUT4 up to the cell surface to get glucose into the cells. Not GLUT1, not GLUT3, just GLUT4. It really looks as though the way the hippocampus helps to regulate the increased demand for glucose when you give the rat or the human a cognitively challenging task is to move that GLUT4 up to the cell surface. And again, notice, there isn't any change in total protein amount. The cell isn't making more protein. It's just sending it up to the plasma membrane. When you put the animal in a maze task, it's sending that receptor up to the cell surface saying, get me more glucose. Very, very specific, very consistent. Spatial working memory, also fear memory. Forming new memories appears to require and cause GLUT4 to go to the cell surface. You don't see that during retrieval. Kind of a side note, it's memory formation, it's not memory retrieval. One of the things we're looking at in the lab right now is why that's the case, but that's kind of a side note. All right, if we think GLUT4 is involved, does it explain the memory benefit you get when you give insulin? So this was a follow-up set of studies. We gave insulin, we showed again, yep, we can boost memory. And if we give a drug, it's called indinavir, that specifically blocks GLUT4, we reverse that effect. So these animals got insulin and indinavir. No effect, no improvement in memory. So just blocking that one mechanism, blocking GLUT4, reversed the effects of insulin. Indinavir, turns out, has a pretty strong affinity for GLUT4, doesn't really block GLUT1 anywhere near as much. If we give a slightly different drug called atazanavir, which is very similar to indinavir, but has a much better affinity for GLUT1, much lower affinity for GLUT4, no effect. So the conclusion is it really is this blockade of GLUT4 that reverses these effects of insulin. And you see the same thing if you look directly at glucose uptake. So these are hippocampal neurons now in a cell dish. When you give glucose, sorry, cancel that. When you give insulin, glucose uptake goes up, as you would expect. Insulin is pushing this glucose into the cells. And if you give indinavir as well as insulin, it entirely reverses the effect. So our conclusion from these several studies, insulin is definitely working in the hippocampus. It's increasing memory. It's increasing glucose usage. And it's doing it via this specific glucose transporter, GLUT4. What about beta amyloid? What about the Alzheimer's brain model? We showed you that beta amyloid didn't affect GLUT1 and GLUT3. It doesn't change total GLUT4 amounts, as you would expect. But it does markedly impair that GLUT4 moving to the cell surface. So in the brain of animals, just given a little beta amyloid, there's less GLUT4 at the cell surface, as shown here. So amyloid is the red bar. S stands for scrambled. This was one of the control experiments we did. We give beta amyloid, except we scramble the order of the amino acids, so the protein doesn't have the same effect, doesn't work. And you can see that looks just like controls. It's got to be the active beta amyloid causing this impairment. Downstream insulin signaling also turns out to be impaired by beta amyloid. And so here's the net kind of summary slide. If you don't follow all those molecular details, you shouldn't stress about it. You should stress that you understand this end result figure, right? This is something that's likely to be important for you. So here is the basic situation in the hippocampus. Insulin comes along, binds to the insulin receptor, activates this downstream signaling cascade, and causes GLUT4 to go up to the cell surface. It moves these glucose transporters from kind of their storage depot inside the cell to be active at the cell surface so more glucose can come in. And lots of good things happen. The animal is smarter, you get better glucose metabolism, everything's happy. In the condition where you have too much beta amyloid, and so you've got these toxic little clumps of beta amyloid running around, they bind to insulin receptors. They prevent this whole cascade from occurring because insulin now can't get to its own receptor. So glucose can't come into the cell. That's why you don't see that dip. That's why when you give beta amyloid, you get this flat line of glucose and you get impaired memory. And so the interaction of insulin and beta amyloid on glucose metabolism, on cell function, on memory, we think is in large part because of this effect on GLUT4, because of this specific glucose transporter as a common mechanism for insulin and beta amyloid. Interestingly, it turns out that probably you get accumulation of beta amyloid long before you actually show signs of Alzheimer's disease. And this is something that's clinically being suspected for a while, partly because it's so difficult to treat Alzheimer's. You really need to go and treat people kind of 30 years earlier and prevent them from developing Alzheimer's. So we did a kind of cool study, I think. We worked with a protein engineer at RPI, a guy called Pete Tessier, to build little antibody proteins that will specifically bind to particular forms of beta amyloid, to the oligomeric form, that kind of toxic form, or to monomeric, the helpful form that's involved in inflammation and so on, or to fibrillar, the kind that's in plaques. So we can design these antibody loops to be very specific for those different forms of beta amyloid. And then we can give these little protein blobs, we call them gamma bodies, to try to block just that particular kind of beta amyloid. The same way we block insulin signaling. We're giving a little antibody-like molecule that's going to mop up that form of protein. What happens when you do that? So if you give vehicle, as you would expect, the diet-induced obese animals show impairment. If you give the form of beta amyloid blocker, that blocks fibrillar beta amyloid, so it acts to prevent plaques somehow. No effect. Kind of what you'd expect. All of this kind of beta amyloid is just sitting there over in a plaque. It's not really doing anything. But if you give the form that blocks the action of oligomeric beta amyloid, now you reverse the impairment. The first time we did this, our treatment here also blocked monomeric beta amyloid. And when we gave that drug, we actually saw an impairment in controls, which is kind of scary. A second better study, we have a form that just blocks oligomeric, and now that causes no impairment in control animals. So the basic result here is these animals are diet-induced obese, they're insulin resistant, but you wouldn't characterize them as having Alzheimer's. Nonetheless, just by blocking beta amyloid accumulation in this toxic form, we can entirely reverse the cognitive impairment. So the type 2 diabetic brain, and this is almost certainly true in humans as well, is explicitly a pre-Alzheimer's state. You're getting that buildup of beta amyloid, probably for 10, 20, 30 years. It's eventually going to tip you over into having Alzheimer's. And so in the middle of these three modulators, right, glucose as a direct modulator of cognition, and insulin beta amyloid as molecules regulating glucose, you've got this fourth molecule, this insulin-sensitive glucose transporter, GLUT4, expressed in the hippocampus, almost certainly one of the final common pathway molecules involved in mediating the effects of insulin and beta amyloid. I should probably say, by the way, that was a pretty intense series of slides. And obviously it's stuff that we care about deeply in the lab and really get into the weeds on sometimes. If you didn't follow all of the details of the molecular pathways, don't stress it too much. You want to get the top level results, right? You want to get the idea that insulin enhances memory by pushing glucose through GLUT4, that insulin and beta amyloid compete with one another and oppose one another, and that beta amyloid prevents that GLUT4 translocation by preventing insulin from binding to the insulin receptor. Turns out there's a third condition, at least, where GLUT4 is involved, as well as type 2 and Alzheimer's disease. There's an interesting condition called HAND, HIV-associated neurocognitive disorder. And if you look at HIV patients, many of them report cognitive symptoms as a side effect or as a symptom of their HIV infection. This came out of basically my graduate student being curious. The whole line of research that resulted in a really cool recent paper came from the fact that he had a mother who's a nurse, and mentioned this kind of odd cognitive finding to him. And he started looking at HIV protease inhibitors. So these are antiretroviral drugs. Indinavir, the drug we saw a moment ago that blocks GLUT4, its clinical use is as an antiretroviral. And it turned out that patients with HIV who were being treated with indinavir developed diabetes, developed cognitive impairments. If they were treated instead with atazanavir, remember that drug that does the same thing but doesn't impair GLUT4, they don't develop cognitive impairments. And so one of the recent advances in clinical treatment that's come out of some of this work is that patients really probably shouldn't ever be given indinavir. They should be given GLUT4 sparing antiretrovirals because it will prevent them from developing diabetes, prevent them from developing cognitive impairment. And like I said, this whole avenue of work and the fact that we have indinavir as a tool in the lab to block GLUT4 really just came out of my graduate student talking to his mother and spending a few hours in the library, created a whole new line of really high impact work. So take part in those kind of random, interesting conversations with people who know stuff, and go to the literature. There's a great saying that you can save 5 months of lab time if you just spend about an hour or two in the library. It's really true. It's always worth doing your background reading and checking to see what you're about to do and what's known before you start doing it. And so, yeah, the key advice from that work is you got to start early. The time to not get Alzheimer's is now. Is go work out, go drink coffee maybe, make sure you're not having too horrible a diet, you're getting good sleep. Diet definitely matters. So we have a study in the lab where one of my undergraduates actually, in these diet-induced obese animals, also gave them resveratrol. Resveratrol is an antioxidant, and she very clearly showed, here are the control animals getting vehicle. She showed this effect of resveratrol may be having some small effect in the control animals. The high-fat diet, so this is orange compared to blue, animals getting the high-fat diet compared to the control diet, definitely impaired. And when you gave those high-fat animals some antioxidants, it's actually the antioxidant found in red wine, you reverse the impairment. So coffee is good for you. Red wine may well be good for you in small doses. And we saw effects at the molecular level as well. One part of diet, caffeine, right? So we wondered what was going on with coffee because there were several kind of anecdotal reports that caffeine appeared to protect against diabetes and maybe Alzheimer's. Was this to do with insulin signaling? What was going on? So we gave caffeine to our diet-induced obese animals. One of the reasons I put this in the slides is because this is entirely undergrad-led, right? So Greg Moy was an undergrad in my lab. This was his honors thesis work. He got a first author publication out of it. He got into med school on the back of doing this work. Really nice study, really clean results. So we had 4 groups of animals: control guys, control with caffeine, high-fat diet with caffeine. And what you see is the animals that got the high-fat diet, as you would expect, gained much more weight. And if you look at their fat pads, those were also markedly increased. But the animal group down here that you can barely see is high-fat diet with caffeine. So this is once weekly administration of caffeine to these rats. Completely prevented the weight gain, completely prevented the obesity in the high-fat diet group. This was really strong confirmation that you're getting a direct reversal and direct blockade of some of these negative consequences of a high-fat diet. We wondered whether it was going to be cognitively effective, and the answer is absolutely yes. So his data showed that you see a marked impairment in the high-fat diet group, but here's the high-fat diet caffeine group. Completely reversed the impairment. Here are the 2 control groups. So you reverse the weight gain, you reverse the cognitive impairment. We then kind of wondered, well, are these guys just really jittery and running around all over the place? No. So if you look at the total number of arm entries, total amount of motor activity during the maze task, there's no effect at all. So it's not that they're hyperactive or anything. It's just that the caffeine appears to be reversing many of the molecular consequences of the high-fat diet. So if you're going to go have a Twinkie, at least have some coffee with it. There are a long list of conditions. Parkinson's, the most recent data are somewhat less enthusiastic. It's not going to hurt, but it may not help Parkinson's. But Alzheimer's, type 2 diabetes, heart rhythm, many of these things are likely attenuated or prevented by coffee. Obviously, the downside is, in some patients at least, it impairs your ability to sleep. Me, not so much. I can drink espresso and then go to sleep quite happily. But if you are particularly sensitive, you can get sleep loss. And one of the reasons to worry about that is that sleep is actually especially important for Alzheimer's disease. Your brain clears beta amyloid, especially during the night. What actually happens is the flow of CSF increases 2- or 3-fold while you're sleeping, and that really helps to clear out waste proteins, specifically beta amyloid. And so one of the big risk factors, along with type 2 diabetes, for development of Alzheimer's disease is lack of appropriate sleep, abnormal sleep patterns, not consistent sleep at night. Sleep is really important for brain health. All right, so kind of summing up some of the lifestyle stuff. A little bit of glucose, a little bit of arousal, some caffeine, all those things are good. Too much of any of them, right? Too much glucose or insulin is diabetes, insulin resistance. Lack of sleep, being too awake, too stressed, too on edge. Too much glucose and fuel, high fat content, you get obesity, you get type 2 diabetes. Too much of anything is really, really bad for you. Lack of sleep, as I mentioned in particular, there's a whole story about the importance of sleep, but one thing in particular is really clear, that you need to clear out that waste protein. You need to clear out beta amyloid from the brain. That happens much better while you are sleeping. If you look at the number of people in the US and around the world with type 2 diabetes, it's massively increasing. The same thing is true for Alzheimer's disease, right? So here we are in 2020, number of people in the US with Alzheimer's. Here's the projection. It's going to more than double in the next 20 years or so. it's already one of the biggest increases in terms of killers. So things like HIV, stroke, we're making good progress in preventing their impact on death. Alzheimer's, not so much. Alzheimer's is causing an increasing number of people to die. What you're getting is a segment of the population that is obese, cognitively impaired, type 2 diabetic, and developing Alzheimer's disease. And both on a kind of personal health, this really sucks level, but also on a society, how do we afford to pay for the healthcare here? It's a really impressively scary trend. Go to the gym, go exercise. It will improve all of these things. Okay, it doesn't improve coffee. Take some coffee, then go to the gym. And actually, that will really help you work out better. There's a reason that caffeine is a banned drug in competition. If you have a cup of coffee and then go to the gym, you'll get a better workout, which will in turn help you sleep better. So it really is important to take care of the periphery, take care of that bag of flesh that your brain is being carted around in order to ensure brain health. That gives you a summary of kind of one of the major lines of research in our lab. Obviously, I find it fascinating. I will be particularly welcoming of questions on this topic for anything you didn't understand. Next time, we're going to come back and change gears to the last major topic of the semester, which is consciousness. All right, we've, as I said, kind of touched on this at several stages. Parasites today are getting to this. But what is in charge in your brain? How do you know? Where is that sense of self? How do we mess with it? Related topics like that. Sorry I can't see you in person. Hope you're all doing well. Stay safe, stay healthy. Put questions in the discussion group. It's right now Monday at 4 o'clock. I'll get this up as soon as I can, so it should be up there at 4 or 5. My apologies again for it being a little late. I will try to get class 23, which is Thursday's class, also up either this evening or latest Tuesday morning. Thanks, see you soon.